Hello students, let's understand what is meant by genetically engineered insulin and also have a look at all the questions that I'm going to put up after this from the previous year's question papers. Alright, so as you know, all people who are diabetic have very high blood sugar because in their body insulin is not produced by their pancreas. Now what do? They have to go for some medication sometimes helps by activating the pancreas to produce insulin and their blood sugar is controlled. For some people, the medications do not work and they have to go for insulin shots. The insulin inside the insulin shot has insulin from pigs and cattle that are slaughtered. Alright, so definitely it can cause some reactions, allergic reactions in some of the patients because they are not human insulin. Therefore, attempts to make genetically engineered insulin was done and it had the challenges that it had many challenges and one of them was that assembly into mature insulin was not possible. To understand this statement, we should know how is the structure of insulin in our body and how is it initially produced. So in our body or the structure of insulin in general, when we talk about the insulin structure has two polypeptide chains. You know what is a peptide chain? I hope all amino acids together form the peptide, and polypeptide usually means many peptides joined together. So, insulin contains polypeptide chain A followed by polypeptide chain B connected by disulfide bonds. This is the final structure of insulin. But in our body, when our body is producing insulin, it is not prepared this way. Instead, it is prepared in this format and then converted into this. So, what do I mean by that? In our body, insulin is initially prepared as proinsulin, which has chain A followed by a C peptide sequence followed by the chain B. After it is prepared, a lot of post-translational changes occur, which we call as maturation, and then it is converted to this format, that is polypeptide chain A, then connection to polypeptide chain B through disulfide bonds. The challenge, as you can understand from this, is that when something like this is prepared, to get rid of this peptide chain would be a problem. Especially inside a vector, you can imagine a chain is there, a long chain structure is there of protein. To cut this off was the challenge that the GE engineers had or the genetic engineers had. So who overcame this? It was overcome by an American company called as Eli Lilly in the year 1983. What did they do? They prepared the DNA sequence or the gene for A chain separately and introduced into a plasmid, B chain separately and introduced into a plasmid. Both the genes coded for their protein. Both the proteins were connected together after extraction by disulfide bonds. This is done by Eli Lilly company in the year 1983. Now let's have a quick look at all the questions that can come from this topic. Try to read the question properly and answer it by yourself. If you're unable to answer, please go back to the video and watch it so that you can answer these questions by yourself. Thank you.